Thank you, Mark. The next talk will be Ann Kwitek from the University of Iowa. She'll be talking about genome editing in rat models.
You did it perfect. All right, instruction, the green button goes forward, and the explosion is the light. Okay, great. All right, I want to thank you for inviting me to come to this really interesting session. I have to give you the disclaimer, I'm not a toxicologist and I don't currently work a lot of in environmental health, but I'm really pleased to at least talk to you about the work that we've been doing. My focus is really on complex traits. So we have heard about how do we address complex traits in the context of exposures and so forth. So hopefully what I go over is sort of an approach that we've used for identifying genes for complex traits and how it might be applied in environmental health. So I always like to say rats, it's complex. And so the topics of my talk is using rat as a genetic model for common complex disease. Some of the approaches that we've used to identify genes for complex traits using the rat as a model organism, and maybe some challenges and opportunities in rat genome editing for environmental health research. So I always have to start out with, why do I use a rat? I'm a geneticist by training, and so people always say, why are you using a rat for doing genetic studies? So the rat has long been used as a model organism, and I don't think I have to really tell you that, or convince you that it's been historically a preferred model or heavily used model for toxicology and pharmacology. There's over 200 years of physiology, pharmacology, toxicology data in the rat model. But you may not know that there's a whole lot of inbred strains that are available in the rat as well. So as of last count in the rat genome database, there's only over 3,500 inbred strains of rats. And really, the history of using them has been to identify multigenic models of complex disease. This is usually capturing population variants through selective breeding, and I'll kind of go through that in just a moment. But the rat is actually a pretty versatile genetic model as well, even without the recent advances in the editing. There are a lot of genome resources in place for the rat. There's over 48 sequenced genomes, different inbred strains. About 200 strains have been genotyped with a high throughput or a high density SNP array. There's a dedicated genome database for the rat where they do a really nice job of curating not only the gene information, but also the phenotype information. They bring in also annotate data from the comparative toxicology database as well. So they try to link, really heavily link disease and phenotype in genome. And that's really what the focus of using the rat for genetic studies has been on. As of about a decade ago now, genome manipulation is feasible. It started out with zinc fingers. The rat model was the first mammalian model to use zinc fingers to knock out a gene. It's moved through talons and CRISPRs as well, and there's a significant number of transgenic or gene-targeted models now. And so I'm going to try to convince you that the rat is a good model for gene discovery and functional studies in gene bioenvironment research. So this is one of the resources if you're interested in seeing if there is a rat model that's been knocked out. The Gene Editing Rat Resource Center at the Medical College of Wisconsin has been very busy knocking out genes that are related more to heart, lung, and blood type phenotypes because that's who funded them, but these are readily available. So if anybody wants to go cruise their website, it gives a list of several different genes that have already been knocked out in various different rat strains. So this is sort of our approach on trying to identify the genes related to complex disease. And my focus is really in hypertension and the metabolic syndrome. So we've started out with animal models that have been selected to have a phenotype. We go through the genetic studies. We use a lot of different genome approaches or different omics approaches to try to identify the variants that are causing the phenotype that we're seeing in these models. And now we've been able to move into doing some genome editing, really to validate what we found by the genetics. So just as a reminder, the metabolic syndrome is really a collection of complex traits in itself, including central obesity, high blood pressure, high triglycerides, low HDL cholesterol, and insulin resistance. And then if you're going to the doctor, you have to have any three of these will give you a clinical diagnosis of the metabolic syndrome. But all of these are complex traits, and they all have genetic and environmental components to these. But really the genetic links that really tie these correlated traits together is really unknown. So we've tried to take sort of a general approach for identifying the genes for these quantitative or complex traits. We start out with physiologically characterizing a bunch of different inbred strains, then selecting a susceptible and a resistant strain to do genetic mapping studies, to map the quantitative trait loci or through genome-wide associations. Go through some other types of genome or omics tricks to identify the mutations by sequencing or looking at gene expression until we get down to sort of that candidate gene that we've narrowed it down as far as we could go, but you still have to prove it. And I think this has always been the issue with using these model systems. If you can genetically manipulate them, you can actually prove what you guess or what you've hypothesized with the genetic and genome studies. So our model that we work on are called the Leon rats, and this is a spontaneously hypertensive rat model. So there was the Leon hypertensive and the Leon normotensive rats that were selectively bred from a single outbred population of the Sprague-Dawley rats. And in fact, there were only 6 rats that started out both of these strains. So basically, you selected the offspring that had high blood pressure, and you breed those together, and you select offspring that have normal blood pressure, and you breed those together. And by doing that for several rounds, you essentially fix whatever causes them to be hypertensive or whatever causes them to be normotensive. So at the end of the day, you have 2 inbred strains, one of them that is the Leon hypertensive, or the LH, and one of them is the Leon normotensive, or the LN. But when these actually were being looked at, now this was back in the late 60s, early 70s. And when the group in Leon started characterizing these strains, they found out that they're not just spontaneously hypertensive. They really have full-blown metabolic syndrome. So the LH rats have high blood pressure, spontaneous. You put them on salt, it gets worse. Their triglycerides are increased, their cholesterol's increased, their body weight's increased. They're less glucose tolerant, and they have hyperinsulinemia. So this is without any kind of dietary or any chemical or anything. These are just susceptible genetically. So this is about 10 years of work in one slide, which is really depressing, but for the interest of time, I'm going to do it this way. So we started out with the genetic studies to try to map the traits onto the genome through an F2 intercross between our normal and our hypertensive rat. And what we found was that one of the chromosomes actually had a bunch of the traits underlying the metabolic syndrome all mapping to the same chromosome. So we thought that was interesting, thinking that this might be a single genetic link that could link these different complex traits within the metabolic syndrome. So this was the first thing, trying to find where the traits map in the genome. But then we actually went to the genome and went back to an F2 intercross and started looking at where's the genetic regulation of the gene expression. And we looked in liver, mainly because of the lipid involvement. And at this point, for RNA sequencing, it was fairly new, and liver was really kind of a straightforward thing to start with. And really what we were trying to do is overlap where we have our phenotypes mapping and then overlapping where we have genes that are regulated by genetic variation. And lo and behold, we found a really nice candidate by just overlapping our EQTL, our expression QTL, with our PQTL, or our phenotype QTL, on this rat chromosome, where we found a single transcript, and this is an ORF, unfortunately. We have no idea what this gene really is, but it is the paralog to the H6ORF52 in the human. This is on rat chromosome 17. So what else we could look at was the genome, sort of the breeding history of these two strains. So both of these strains were sequenced. We know the sequence of their genomes. And because they were derived from the same 6 rats, basically, they're very, very close genetically. And so we were able to look at where they actually differed, with the hypothesis of where they differ would be where their genotypes differ is where their phenotypes differ. And when we look at chromosome 17, we see that there are very small regions of high sequence variability with very large interspersed regions that have essentially no polymorphism between these strains. So it turns out that our C17H6ORF52 is in one of these haplotype blocks of high genetic diversity. We've shown that it is genetically regulated by those sequence variants, and that it overlaps with this QTL for different traits underneath the metabolic syndrome. So there we were. Now what do we do? Well, fortunately, we were able to get a knockout rat using CRISPR through the Medical College of Wisconsin. They actually were taking applications for people to just nominate a strain and a gene to be knocked out, and they'd do it for us. So that was awesome. And I spent a lot of years at MCW, so they knew my name. So when they came in, I probably was able to convince them that even though we knew absolutely nothing about this gene, that it was worth doing this knockout. So we sent them our strain of interest. And what our strain of interest was an LH congenic strain that had a normal LN allele of this gene. Now, what I don't want to go into, but I have to go into a little bit, is that sometimes a single gene is not sufficient to overcome the homeostasis of a system. So we made this congenic, hoping that putting the normal allele on this LH strain would make them less hypertensive and make them leaner and so forth. Didn't happen. It was not sufficient to actually reduce this susceptibility for being obese and hypertensive, because there are other genes that in this background that have this susceptibility. Regardless, we decided that we were going to knock out the normal allele in this congenic strain, and there was also a reason that they bred a lot better than our parental LH rats. So we sent this congenic and had this gene knocked out, and they gave us back 2 different mutant lines. They were able to give us one that had a 5 base pair deletion in exon 2 of this gene, and one that has a 1 base pair deletion here, followed by an 11 base pair insertion, so it was an overall 10 base insertion. So both of them caused frame shifts and truncations of the putative amino acid sequence of this gene. Again, here this is just called RGD because previously it just had a transcript identifier that was assigned by the rat genome database, so it was RGD 1562963. Now it's C17H6ORF52. They're both mouthfuls, and they have it doesn't give you any hint on the function of this gene, but we at least could look and to see if there was actually a phenotype with this gene. So we got these 2 mutant lines, and we are still characterizing them, and this is still not published, but what we are seeing is that it looks like the knockouts of this gene have an exacerbated salt sensitivity phenotype. So their blood pressure gets higher when you put them on salt, but if they are on the normal salt, they are not any different from our wild type strain. Keep in mind our wild type strain is actually hypertensive. So it looks like this gene may actually have some sort of a protective effect so that when you knock it out, the susceptibility actually gets worse. So in the Leon hypertensive rat, this may actually be a protective gene to keep them from being so obese and so hypertensive that they stroke and die. That's my take on it right now. But at least in the females, it looks like they have an exacerbated blood pressure. In the males, it looks like their cholesterol levels are also exacerbated. So I'm not going into too much more detail on that, but what I'm going to tell you is some of the challenges and the opportunities that we found with this strain. There aren't that many rat knockout strains yet in the literature, but what we found as far as off-target effects, and we did not specifically look at the genome for the off-target effects. That would be something we should maybe do. We looked phenotypically because that's what we had was the animals. So because we sent this congenic strain to them, we knew the phenotype of the congenic. And so when we first started looking at this, they gave us a handful of homozygous mutants. But what we saw was that there was a nice significant difference in body weight between our parental congenic strain that we sent them for targeting and our knockouts. However, when we finally got the animals and started breeding them, we saw that both the wild type and the knockouts from this line are now have lower body weights. So the body weight phenotype is not a genetic determinant from our gene. I think it must be an off-target effect, but I don't know that. What we're trying to do is back cross it away because we're trying to study our gene. We haven't actually looked to see what the other gene might be. So that's a complication, but fortunately we can probably just keep back crossing. We're in the 5th generation now, and so as we keep phenotyping, hopefully that will go away. So that's one of the challenges we came into with the CRISPR off-target effects. I'm sure better CRISPRs and Cas enzymes are happening now to prevent that from happening, but that was our experience as we got these a couple of years ago. Also, I want to say a little bit more about the inbred models. So I'm working with inbred models. I've always worked with inbred models. As a geneticist, that's what we preferred working with, right? But of course, we have to keep in mind that inbred models don't recapitulate a population within the species or across to translating to human. They're great for identifying genes and causal variants, and we use them all the time, but they're not ideal to translating a population for risk assessment or potentially for regulatory purposes. And so, you know, having genetically tractable population models are quite needed, and you heard quite a bit about that with the collaborative cross and the diversity outbred models with the mouse. The one thing I can say is that, you know, when you're looking at a causal variant in an inbred rat, it's just like in humans. There could be a common variant that you see across other strains of rat, or it could be a rare variant that you're seeing in just your own strain of rat. In our case, it's pretty much only in a couple strains, and the other one being a salt-resistant model of hypertension. So we heard about the diversity outbred mice. I'm going to talk just a bit about the heterogeneous stock rats because it is another resource to do a population-based genetic study. And so we could potentially be doing mapping in the outbred populations of the HS and the diversity outbred. You can map these traits at high genetic resolution to a few million base pairs or maybe a single gene. The heterogeneous stock rats have shown numerous segregating phenotypes showing that they're actually susceptible to metabolic traits, cardiovascular traits, behavior, and addiction. And so if your model, if the rat happens to be a more appropriate model for your question, it's a good way to go. It's great for mapping, but your causal gene identification is still a challenge because you're working now with a population. So one thing that you may be able to do is you map your genes in exposed rats or in the exposed mice that are a population base. Then you can informatically go in and look to see what is the identity of the ancestral founder, and then go back and do your gene editing in that strain. So with that, I will summarize that the rats are really good models to study complex traits and gene bioenvironmental interactions. Gene editing is feasible and is a necessary tool in the rat to prove causality. And the use of inbred or outbred rats really depends on the question, but both can be genetically tractable. And I'd like to thank my group for their work that they've done on our rats. Thank you.